Welcome everyone to the Evidence Reasons Academy. I'm Sal Cordova, and I'm joined by my illustrious, distinguished colleague, Rebekah Davis. Some people have called us the Power Duo. Yes, that was a new label, and I'm appreciative. I certainly feel that uh, she's one of my favorite co-hosts. We have great shows. We also have had one debate together, and right now we're off the debate circuit, but we've had a lot of fun. We are preparing to hopefully interview Dr. Scott Minnich, who is a very respected researcher at University of Idaho. He was a Princeton postdoctoral fellow. Scott Minnich was also notable for his participation in the infamous Dover versus Kitzmiller intelligent design trial. Scott Minnich appeared in Illustra Media videos advocating intelligent design, and he conducted experiments that showed Lenski actually made mistakes in the long-term evolution experiment. So that's what we're going to talk about. And we are just going to, Scott Minnich had a video that he did with Jonathan McClatchy, March 7th, 2017 on the Apologetics Academy. It was extremely technical. We aired it here in a rebroadcast on this channel. And Rebekah and I are going to kind of review what we saw in the things that confused us and the misunderstandings we had. I will tell you, I got a little bit cross-eyed when I started to see some of the terms. I'm just like, I study eukaryotes. I've never seen that term, like polycistrons and operons. That's tends to be very represented in prokaryotes, not eukaryotes. And I'm just like, oh boy, there's going to be a big learning curve. I think there are some questions that are raised. Interestingly, some evolutionists have and I kind of saw this. They said, well, Scott Minnich actually proved that evolution works even better than what Lenski argued. And I'm just like, well, there is that argument has to be addressed. And so when Rebekah and I called this meeting, we actually didn't know specifically what we were going to talk about, because we didn't know in advance what sort of problems and complications we're going to run into. And that's still kind of the case. So this is going to be a free-form discussion. And we agreed that we're going to try to iron things out. I would, because I have some biology background, if I could answer a question, I will. But it might also be that, hey, we're going to have a list of questions we can't answer among ourselves. We might have to present them to Dr. Minnich. We also have concerns that, does further work have to be in this area, or do we have to rethink things? Also, there's a time constraint. We don't know how long Dr. Minnich will be willing to grant us. I know he has updated presentations of the same topic since the 2017 version. I've been privy to the more recent versions. He has emailed me and expressed some of the things that he's improved upon, that he said he wished he hammered harder. I am hoping that his protege, Dustin von Hafwegen, who's now a professor, and he was an NIH fellow or researcher for a while after getting his PhD, I'm hoping he'll come on this channel. But Dr. Minnich said, well, Dusty's in the middle of a relocation, moving houses, and he might not talk to you for a while. Dusty and I are on good terms. We see each other at various conferences, and he's complimented some of my work, which I was very appreciative of. So I'm hoping that we'll be able to have Dusty here and we could talk about the technical details. Any of you who have actually looked at the paper, I looked at the paper and I was like all cross-eyed. I couldn't get through the first paragraph. They use such, okay, someone who studies eukaryotes like me was totally lost in the prokaryotic world, microbiology, E. coli, all the notation and terms are just like, I have no idea what you're talking about. So I'm hoping that maybe we could get a hold of Dusty. Rebekah, if you have any opening thoughts, I did want to, as you might be sharing your opening thoughts. I'm going to pull up a slide, and you know which one I'm going to pull up.
The prokaryote, eukaryote differences?
No, I'm going to show that picture of Zach Blount versus Dusty, and they showed the mountains of Petri dishes and said, look, it took them 15 years and 33,000 generations. It took Dusty just a few Petri dishes to do the same. I want to pull that up. I thought that was really.
And maybe I'll kind of tell the significance of this for those, I'll just do a quick recap. So the Lenski experiment is often called like the, best experiment that's done on evolution. It's definitely the longest experiment that's been done with evolution. And it's just, they've been watching E. coli for more than 30 years and watching how they change, changing them from, Petri dish to Petri dish and kind of watching the development. And there have been some changes. They got better at metabolizing glucose. They, got bigger. And then there was one development in particular that was pretty astonishing. It was the one population of the E. coli developed the ability to metabolize citrate, which was not previously. And one of the questions I have, I'll ask later, was about the anaerobic versus aerobic. But from what I understand, they could not metabolize citrate. And then they were able to metabolize citrate. And that was hailed as, see, we can develop new abilities through evolution. And this is a great success. It shows how evolution is able to change organisms to develop new abilities. Well, what Scott Minnich did was he reproduced this with a slightly different variety of E. coli. And he, within what took like 12 or 15 years of evolution in Lenski's lab, Scott Minnich and his team were able to reproduce this in just like a couple of months or less than a couple of months. So days to months rather than years. And he reproduced it again and again. So what it kind of showed was that E. coli, actually, it's not that hard to get them to develop this ability. Now, what's interesting is that, and this is where, Sal, I'll ask you to correct me if I'm wrong. This is what I'm checking against you to make sure I understand it properly. So what he was showing was that, there are some things that are interesting about the development of the ability to metabolize citrate. It doesn't start as a natural ability, but it develops with a couple of things: gene duplication and then amplification. And there was one other thing that had to happen within that to make the E. coli metabolize citrate. But he was able to
promoter capture.
Okay, so I don't know what that is, so maybe you can explain that later. So, but he was able to consistently get the E. coli to develop that ability. So is that really evolution? And what Lenski's lab is promoted when they said, hey, this is evolution, is they talked about how this is like, I think they called it like historical contingent, where it was contingency. Okay. So basically what Lenski's lab was saying is that it took many neutral mutations occurring before it was able to develop this ability. So the entire thing was based on the neutral mutations accumulating before the mutation that allowed this thing to happen. And that is what is presented as the main way that evolutionary change happens, is that you've got neutral mutations that accumulate and they're supposed to, eventually produce something good. which if that's true, that is a big win for evolution, right? That's a demonstration of the power of evolution. But what Scott Minnich's team showed is that their E. coli did not have to undergo all of those neutral mutations. Their E. coli, without those neutral mutations, was still able to make this jump and do the gene duplication and everything required to metabolize the citrate. And so those neutral mutations that happened in Lenski's lab are irrelevant to the process. And so that's what I understood from that. Is that a good summary, Sal?
Yes, that's a good summary of the 2017 presentation. I'm privy to some stuff which Scott has written to me and said, I wish I had added these points more forcefully. When way back in 2017, before he had a back and forth with the man himself, Lenski, he didn't say certain things, but in the process of that, he's like, he caught Lenski saying some things that, you know, Scott looked back. There's a fun, I don't know how much, he said it's probably in his responses way back in 2017 or whenever he and Dusty did this, but he said he wished he emphasized the point more. Lenski picked the one strain of bacteria that was defective. That's why it took it 15 years. The more common wild type, I think it's E. coli K12. It's more representative of what's out there. If Lenski had picked that one, he would have been able to evolve this much quicker, because it had more capability. And this is what I want to clarify with Dr. Minnich, is that actually it had to break one or two more things to kind of compensate. And to get those mutations, that's why it took about 15 years. It took a while for it to actually break some things. And I have the email here where he actually describes, and he's pointed this out. He said he wrote this in his responses. So this is the picture I wanted to show. Okay, on the left is Zachary Blount, Lenski's assistant. On the right is Dustin, or we call him Dusty. So Zachary Blount is Lenski's author and assistant, and Dustin on the right is Scott Minnich's assistant. So all those petri dishes, those mountains of petri dishes, is how many it kind of gives a feel for how many generations and how much effort and how many years they had to keep running this evolutionary experiment to get the feature. The main reason it took so long is there was a defective E. coli. Why don't I, let me try to call up the actual name of it rather than being vague. And Dr. Minnich has the name here. Just give me one moment. Let's see, the name of it is it's DCUS. DCUS. Scott said Lenski used E. coli B, that's the name of the strain, and it could not grow on something called succinate as a sole carbon source because it had a 5-paired deletion in DCUS. And so to evolve this, it had to actually kind of disrupt another gene called DCTA, and then it enabled the path. But if Lenski had chosen the more common E. coli, which is K12 wild type, it wouldn't have taken 15 years to evolve this. So, okay, go ahead.
And from what I understand, all of the changes that took place in the E. coli before the metabolized citrate, they were losses of genetic information and ability. And so, and then with regard to the change of the metabolizing citrate, it's a change that one could call it new information, but what Scott Minnich says is he doesn't count that as new information because it's just a duplicate, it's gene duplication and amplification. But what evolution needs to show is that like what we need to show to make evolution a viable theory is that this information has to originate from somewhere. There has to be some source of information for organisms where that, then, yeah, they can duplicate information, but where's the original source? And so that's what he's pointing out as one of the failures of this, is that we're still not seeing new genetic information arising. Even if we, look at what's happened in this experiment, it's, still just a copying of information that was already there.
Okay, so I'm going to deviate from some of the ID proponents and their standard line about information increase. The way it's framed is like someone saying you can't win money in a casino. You can, even if you're not a skilled gambler. You can go in there, you might end up with a little bit more money in your pocket than you started with on one day. But over time, because of the law of large numbers, it's going to erode unless you're a skilled gambler like I once was. But I got kicked out of the casinos because I was a skilled blackjack player. I'd be hesitant to say that there's never an information increase. I would just say, okay, the first thing I'll point out, one of Lenski's lines started to lose genes. And I specifically asked Scott Minnich in that, some people have that preparatory video. You'll see me way back in March 2017 when I was just a beginning biology student, even though I was already an engineer and physics student. I was beginning biology student. And I asked him, what happens if you lose a gene? Can you recover it? Scott said, no, not unless you have other bacteria around that can kind of, they have copies and you can have a horizontal gene transfer. Otherwise, it's gone forever. And so in one of Lenski's lines, I keep quoting this, it says, fitness increases despite sustained, genome decays despite sustained fitness gains. Genome decays even by evolutionary standards, that is a loss of information. You're losing genes. And I was just quipping. I said, okay. I said, Zach or anyone else? And Zach is at the same university as Lenski. Not Zachary Blount, but Zachary Hancock, another Zach. So there are two Zachs in University of Michigan. I said, Zach, how are you going to evolve a microbe into a human being when you increase evolutionary fitness by deleting genes? So I wouldn't necessarily hold up Lenski's long-term experiment. He has these citrate mutants, and some of the best performing ones are losing genes. So I say, okay, I'm going to grant you a fitness. I'm going to grant you information increase, but why don't you tell the whole story? You have a lot of lines that are just losing genes like crazy. They're reproducing like rabbits, or really more like bacteria. Like rabbits as a figure of speech. They're just reproducing like rabbits, and they're losing genes. So, I mean, that's not very balanced to say you had information increase and then you ignore all the genetic losses in the line, in some of the lines you evolved. So I'm just gonna throw that point out. I'm willing to grant a little information increase, but over long term, I'd say it's gonna be deterioration. We have evidence of that. I remember sharing this with people and they just started laughing. People on our side of the argument, they're just laughing. I said, Lenski could not have delivered a better title for a paper because he was co-author of that paper. Genomes decay despite sustained fitness gains. I'm just like, okay, why don't you advertise that? How about a nice big breakthrough that you have an experiment that shows how natural selection accelerates genomic decay? I'm sure, I wonder how much press you're going to get over that. You have This is like the casino, the guy who lost all his savings, he's going to brag about the few times he won.
Yeah, and the thing that I noticed about that experiment is that I think it's at like 75,000 generations right now. I could be wrong, but because I didn't look at the latest, but at least when you guys were talking, I think it was at
65,000.
yeah. So in that many generations, these small changes that have occurred. Now, one could say, well, it's no longer an E. coli because it can metabolize citrate, and that's what makes an E. coli an E. coli. But you've still got essentially the same organism. And so, why is it that in this long term evolution experiment, we can't get an organism to change more than in these very basic ways? And yet, if we took, 65,000, 75,000 generations of humans, and you try to think about the changes that would have to occur in humans in order to get, the last common ancestor or whatever, if you think about the number of changes that would have to occur, it's like, wow, actually what this experiment is showing is that organisms are quite stable. And we see that all over, and people don't want to talk about this, but you've got organisms that you can find in the fossil record that are essentially the same as they were supposedly millions of years ago. And so what about all the stability of organisms? That's where can we actually see these dynamic changes occurring in organisms? And we don't have the experimental evidence for it, so it probably should not be promoted as something that can happen if we have not had experimental evidence that shows it.
Now, see, unfortunately, and this is something collectively, myself included, there are, if I said, hey, look, I can improve the performance of a car, or I could change a car. I could just take a hammer and I'm going to hammer. And I could say, hey, look, a hammer can change a car. Fair enough. But would you say the process of hammering the car is how the engine came about, or all its novel innovations since the early cars? It's like, that's ridiculous.
Right.
There is a parallel. So I mean, the thing is, they can demonstrate one class of change, and I say class of change. You can't extrapolate that as showing that it's going to create other classes of changes. Okay, the obvious stupid stuff that I just can't believe no one's seeing this. You can't be deleting genes saying, look at natural selection deleting all these genes. It's so reproductively successful. Therefore, you're going to evolve a human. I'm just going to laugh. I'm really, do you not see the problem? And I've cited many papers. The dominant mode of reproductive success is gene loss. It's not just restricted to bacteria. It's across all species. I'm like, guys, okay, that's like a really basal problem. You can promote all your natural selection examples you want, but it's not exactly being genuine now if most of your explanations are gene loss or disruption. So you may not totally disrupt the gene. Some other things you could do is alter it, weaken it. And that's why I was laughing. This book, this New York Times bestseller, said survival of the sickest. And I said, this is not what you want if you're trying to evolve complexity. Now, the problem with this E. coli experiment, it's more subtle. There is a distinction between a promoter and a gene. A promoter and a gene.
That's what I wanted to ask you to explain. Can you explain and show what happened?
Okay. And anyone in the comment section, except the people I banned, I'm sorry, who I got fed up with, can correct me if I'm wrong. So this is just from a random Google search. I said promoter and gene. So just kind of the basics. The DNA provides the gene part of DNA. So a lot of the DNA in a bacteria is genes. In humans, only like 2% of our genome is actual genes. So what is a gene? A gene generally there's some kind of, exceptions to this, but it generally is a template for a protein. In eukaryotes, it's more complex. It's a template for some of
Excuse me.
non-translated RNAs. We don't need to go there, okay? But just think of just kind of the most basic central dogma of molecular biology, approximating it. The gene is what makes the protein. So when you say the CYT plus, the CYT gene, it's going to make the CYT protein. It's the blueprint. The promoter part. So this is all DNA. So, but there's a part of the DNA that's gene, part that's promoter, and part that's other stuff. The promoter region is that switches off whether it's going to express the gene, because there has to be a way for the cell to say, look, I'm going to make this much. I need more of this kind of protein to build all my body parts. But when you make enough, you switch it off. So the promoter is basically the switch. So what can happen is, what happens if you duplicate the promoter in a different region? You can actually get it to, so there are certain conditions that will activate the expression of the gene. Do you have any questions here so far?
No, this is great.
Okay, so if I disable the promoter region, for example, let's say I totally mess it up. So what happens in the promoter region is other proteins, first of all, have to dock there and say, okay, the proteins decide whether, okay, if I dock there, I'm going to start the transcription. Or if I dock there, I'm going to inhibit it. There's a whole orchestration of what happens. But all the computation will happen. So what'll happen is these molecular machines will assemble at the promoter region, and then they decide whether they're going to turn the gene on to start expressing or turn it off or block it. I'm probably just totally messing this up, and this is a good Dr. Minnich question. Is like, did I understand this? And so what can happen is like, all right, let's say we accidentally copy this promoter region that's associated one gene and copy it into a region into a different gene area, into a different area. And now all the machinery that was used to work with this gene is now going to be assembling to this other gene, and it'll be turning it on and off. So it's basically switching the gene on and off. So it's very important then to make a distinction between making a new gene, copying a pre-existing gene, and then also the promoter regions. So I don't quite know. I don't have any analogies off the top of my head, but
okay, but that repressor part that you showed, that is like normal, right? Within the process, like there's a repressor to stop the copying when it needs to be stopped. Is that correct?
Yes,
okay.
You can repress, you can promote.
Okay.
And it's kind of funny, the repression happens in the promoter region. Okay, so there has been there's some evolution of terms that, it just kind of got fixed. So when you want to repress a gene, you actually have to go to the promoter region and put a repressor machinery. So one of the things that will start transcribing the gene, and I could see it here, is this RNA polymerase. It's a machine. And one way you could prevent it is you put in the promoter region this repressor. It's another molecular complex. So the importance of the promoter region is, it's like saying, hey, somehow the cell computed that we need more of this material. And that's still kind of mysterious how this is done. And it's sort of simple, relatively speaking, in the bacteria. Compared to eukaryotes like humans, it's just insane. It's so hard to characterize. So the promoter region says, hey, I need more of this particular gene. And it's like, well, the machine will say, well, where will I find that gene? It's like, well, the promoter region has the sequence that's basically like an address. It's a postal address. So think of the promoter as the postal address. This is the area where you're going to regulate this gene. So promoter region is like an address.
Okay, so what happened with the E. coli? So is it that so there was a gene duplication,
Gene
and that was just
duplication.
accidental, as far as we
Yes.
know.
Or
And then,
we can say, let's even say it's purposeful. It doesn't matter. It duplicated it.
okay, yeah, that's what I was going to ask. Because could this be, since Scott Minnich was able to reproduce it again and again, was it really accidental? Because I mean,
Okay, so okay, there's some, and I'm happy to see this. There is some sympathy in the ID community that some mutations are directed. Even Dr. Sanford recently said they are now willing to say they think that sometimes an organism will actually try to reprogram its genome a little bit. There are limits to what it can do, and I'm totally in favor with that. There is kind of like a forbidden thing you do not do in computer science if you're a software developer, is make self-modifying code, but you can do that. If you're a really gifted programmer, you can do that. So I think you can make I hesitate to call it real information increase, but it's like, look, guys, I'm not beyond the idea that genomes can actually learn from their environment. Our brains can learn, the genomes can adapt to their environment. God may have put some intelligence in it, limited capability.
well, we know there's plasticity in other parts of organisms. Like there's plasticity even within the phenotype. There's plasticity. I mentioned the lizards and their cecal valves. When they change their diet, they get a new organ that pops up. And it's like, so that's plasticity. And then when their diet changes back to only vegetables or or to only meat, then that feature goes away. So that there's plasticity within organisms in other ways. So it makes sense there would be plasticity in the genome as well.
So that's why I'm kind of resisting. It's like there's no information increase ever. I'm just like, no, I wouldn't go that far. That's you're going to get slaughtered if you keep, because they're going to keep coming up with these examples, because they'll say, hey, look, Scott Minnich talked about a gene duplication, and and it's like, okay, look, we can bypass all that. I'll grant you a little bit because I have the trump card, so to speak. I'll say, look, you can have your little puny gene duplications. Why don't you just fairly report the fact of all the gene loss in this Lenski line? I'm like, okay, I'm going to win this argument because it's like, if you do the accounting right, this is a disaster overall.
And I mean, you've also got the trump card of, it's fine to duplicate a gene, but you've got to get the original to come from somewhere.
You got to get the original. It's not going to do you any good if you're losing genes.
But Sal, can you clearly say what happened with the E. coli very concisely? So there was what happened with the promoter? Okay. What happened with the...
All right. Short answer, no. Short answer, no, because I'm struggling.
You can do it.
Okay. The short answer is not because I'm normally long-winded and I can be concise. It takes a lot of effort, but I am a little bit concerned that I'm not even understanding this completely correctly, okay. But I will do my best.
Okay.
I will do my best.
So grace from the viewers, If Sal makes a mistake, we're happy to correct it. Okay.
Right. So and this is why we're begging. This is why we wanted to go through this. And so when we go to Dr. Minnich and ask him questions, we're not just asking totally uninformed questions and showing we didn't even give a first order try. We can serve him though by saying, look, if we want to clarify this because we want to promote your work accurately. He'll be very generous if he can squeeze this in. He has a lot of people working for him. He's a very respected researcher. So I'm very grateful I was able to get him to at least tentatively agree. So, but first off, think about that promoter. This is the switch that turns on and off. Now, if I went to my household and I started, okay, like I could, not that I really would want to, and I've sort of done this, if I've taken one switch that controls this light and then just kind of rewired it to control another light, you could do that. This is sort of what's happening, is that it got rewired. So you still have the genes there, but all you're doing now, okay, so you have all this complex machinery, like we call the genes, or like say the computers, and I'm just going to rewire the outlets and the circuit breakers. That's not anywhere near as complex as like rewiring an entire machine. So I'm going to show this diagram here from the 2017 lecture. And
Okay, so the top one is the regular, and
let me show you the other. This is a better diagram. This, let me see, which one is Lenski's? Okay. So this little MK, that's the original promoter region, MK.
Wait, first tell me what each one of these things
okay, all right. So, okay, you have this
quickly, like, yeah.
Okay, so anything, okay, I wish they used different symbols, but basically this is a gene.
Okay.
That color, and then this is another gene. And these genes are side by side. And I think this is a RN something. I wish they didn't call it RNA. It could be another gene or something. That's what I need to ask Scott Minnich, or is that an MA? I think it's
like MA.
Okay. And then this little gray box is the promoter, I believe. And this is why I have an issue with the diagram. And this is why I'm going to say, Dr. Minnich, what is what here? So we do have a promoter, and when
so it looks like the Lenski, I'm trying to figure out, so it looks like the bottom one is Lenski's.
Correct.
Long-term evolutionary experiment at the bottom.
Right.
And so the first one, that WT must stand for wild type. So that was what Minnich started with. And then that's what Minnich got, I guess, the progression.
It's a little more subtle than that.
Okay.
It's a little more subtle than that. It turns out that for this section of the genome, and there's other sections that we're not showing, and that's very important. You could say that Lenski and Scott Minnich started with this, but there's a part of the genome he's not showing, which is that DCUS.
Okay.
Lenski started with a defective DCUS, which is not depicted here. So what Dr. Minnich was pointing out is, if you didn't have that defective part that Lenski had, all you really had to do is take this wild type, and then you duplicate. You could see he puts a little promoter in here, this INSL1, and that'll do it. Or there's another one here where he may put it here, and he could explain this more in detail.
But wait, Sal, clarify. I need the clarification here. He can put it, but this is not happening by him putting things, right? This is happening.
It's just randomly happening. So I'll show you what's really embarrassing is that in one line, the promoter region ended up here. In another line, the promoter region ended up here. And he had so many examples where it's just kind of, you could be inserting these, redoing some of these promoter regions, and it was giving you the same effect. So maybe one way to describe this was there only one road to Rome, or were there many roads to Rome? And it turns out there are many roads to Rome to get the same effect.
Now this brings up another question really quick. This SIT-T section. Now, what I was thinking I was understanding from the thing is that there was an ability to metabolize SIT-T in an anaerobic environment, or was it possible in either aerobic or anaerobic?
It was possible to do it in an aerobic
environment, but what changed is that it had the ability to metabolize SIT-T in an anaerobic.
Right.
Okay. So it already had a capability. It just needed a little adjustment.
Yes.
Okay.
And Dr. Minnich wrote me, he said, I want you to study that citric acid cycle, because every creature can digest citrate. They're just a few things. And that's what I'm sorry to be anal about this, but since Dr. Minnich wrote me, he said, before we interview, he said, I want you to study that citric acid cycle. And I was kind of embarrassed because I did study it in biochemistry. But you saw it, him referring to it in that 2017. So even we humans, okay, we humans at some point in our metabolic cycle have to deal with citrate. So it really didn't have, it wasn't that our body chemistry cannot handle citrate at all. It's the fact that when it's out in the environment, to get it within the cell in a way that it can process. But okay, so let's look at the citric acid cycle. Oh boy. I'm going to see if I could find one that actually uses the word citrate here. And I'm going to embarrass myself. There's so many biochemists that understand the terminology. They could just look at the chemical diagram and tell me I kind of need the names. Let's see if I could find this. And if I don't find it, I apologize to the viewers. And see if I can find it.
Well, if you're watching this on Sal's channel, then it's going to be extended because Sal never edits. So you'll have to watch Sal searching for the thing. But if you watch this on my channel, I will cut all of this out. So you can watch a condensed version.
Yes, and I'm getting a very hard time getting like really nice graphics. So okay, so let me show this, and the viewers who know better can correct me where I'm wrong, but here it is. Look, most organisms have something called a citric acid cycle. Krebs won the Nobel Prize. Took him a long time to figure out this, and you can clearly see it says citrate. Citrate.
Now, what does this mean? The Krebs cycle. What does that mean?
Oh,
that
my
means
goodness,
nothing to me. I don't know what that means.
Oh, okay. So why don't I read the Wikipedia article?
Oh, no.
Okay, so
I need 2 sentences. 2 sentences.
Okay, to metabolize our food, we need the citric acid cycle.
Okay.
That's it. For us
so
to
the Krebs cycle is the citric acid cycle.
Right. It's part of, I hate to call it that digestive process, because some people anal in terms will say, well, that's not really it. I'm like, okay, you take in nourishment, you have to convert that food into usable stuff. Part of that involves citrate being processed at some point. You process citrate at some point. Now, there is some subtleties that Dr. Minnich will point out, as to, you can't say we don't deal with citrate, even us. Even a bacteria that is not a site, cannot live off of citrate. It's still processing citrate. It has a lot of capability to process citrate. I think his point is it doesn't take really a lot. You just have to have the right switch. So that's when I was saying I could move light switches and rewire. All right. You have all the machinery. You're just kind of crossing wires, so to speak.
Okay, so what happened in the E. coli? There was a gene duplication. How did that happen?
Okay, so for some reason, we don't quite understand why entire genes can duplicate. We know that when there's a copy error, it could be just segments. A copy error can sometimes end up making 2 copies.
So which gene duplicated? Can you pull that thing back up?
Sure. I'm a little slow here. Oh, here. This is good. Good question. So just also for my benefit, when a cell is dividing, it has to duplicate its genome. So it has to duplicate the entire genome. sometimes in the process of duplicating the entire genome, it will duplicate individual genes, which is kind of interesting in and of itself. I need to point out one thing though. Scott Minnich specifically pointed out when you have duplicated genes, that means it's having to copy more information. Do you remember the Spiegelman experiment where the genome kept getting smaller and smaller? Because okay, if you have technically, if you have a duplicate of something, it's like, why would I have a million phone books? One will suffice, right? You're having, there's extra load. So he said specifically, duplicated genes are unstable. They could be selected against or drifted out. He didn't use the word drifted out, but I sort of think that's what he meant. So now
they're not going to be as good at reproducing because now they have to reproduce more information.
More, right.
Yeah.
Okay. So even just one extra gene, we think it's going to be hard for it to be stable in the population. And so let's look at this diagram down here. This one right here. This is one of them. You can see it says SIT-T. The little you start with just one orange blob, which is the SIT-T gene. Now you have two copies of it. Okay. That's a gene duplication.
And so did it copy like all the SIT-G, SIT-T, MA? Like was that all?
Yeah, yes.
But that was all part of the gene, the same gene?
Right. It copied. Okay. You could see this is an entire duplication here.
Okay.
You can see like the yellow. So it duplicated the SIT-G, the SIT-T, this thing that's either MA or RNA, and then the MK, and then the YDR.
Okay, and then so what did he mean by it amplified it?
Okay, amplifying means if you have two copies, it's going to make, okay, if you have two copies of the gene, it's going to tend to make twice as many copies of the protein.
Okay, so
it's the way the machinery, it's just the way the machinery works. One way you can make more of something is to have more copies of it. That's all it is. So
okay. So it's not a second thing that happened. It's just that's what naturally comes from the duplication.
Correct. So we do know, it seems one of the natural okay, I'm going to be a little philosophical here. One of the ways that I think the designer has enabled creatures to adapt is that if they really need something, why don't they just have more duplicates of the gene? We could either, okay, so maybe as an analogy, okay, so when you have like a speaker on, in its certain loudness, and you just crank up the volume, you can adjust it within the range of that speaker. But if you really want more loudness, you get two speakers or more. It's really not very sophisticated. I mean, conceptually, it's not. It's kind of the same thing there. There might be a point you could, it's just going to be, you can make much more of the protein just by having a duplicated gene. So it's kind of like an extra speaker to amplify. So that's all it is. He said that's really the first step. And it does help it a little bit. And then there's a phase where there's this promoter capture. It's really more of like rewiring. And a promoter, again, that is a duplication event. That is not, okay. And maybe one thing to bear in mind, and I'll show this diagram again. When we're duplicating parts of the genome, and we just happen to take a piece here and then duplicate it elsewhere, that is not as fantastic as actually creating a new gene. And that's why when I debated RN Raw and I said, look, and he said, there's no effing common ancestor of all proteins. It's very much difficult to just take an arbitrary sequence and just start to create a new protein family. And I tried to show that diagrammatically. It's not very hard by comparison to duplicate pieces here or there. If you have a copy error, you could just like say, like in this case, you see it says the tandem duplication 5,834 base pairs. So that's 5,834 DNA elements. We call them base pairs, but That's how many letters of DNA just got duplicated. That's not really particularly spectacular. If I were to modify, if I wanted to say, like, say, make a topoisomerase from scratch, I would have to have first start off with a string that would be, say, 4,500 DNA elements. And then I would have to modify each and one of those individually. Then I could make a brand new topoisomerase. This is copying something that pre-exists versus, modifying 4,500 nucleotides to make a new gene. That is
Okay.
that's orders of magnitude more difficult.
So what does it mean, promoter capture? What is it showing here? The promoter is the gray thing. So what is it
Yes.
showing us that what is promoter capture? What does that mean?
Okay, so what happened is I'm going to go with the Lenski one because that's the one I sort of understand. And caveat, Dr. Minnich will clarify this diagram, God willing, when we talk to him. So this is what I think is the promoter here, where it says RNK sit G or MK sit G. This is the promoter region. So in the original, you don't see that. What happened is this kind of got copied, a piece of this got copied into that, which is kind of funny. And it ended up becoming a new promoter. So you could see this is the way it looked before.
Okay, I see. Yeah.
And remember, a promoter is not very sophisticated. A promoter is not a gene. It's like an address. A promoter is. Have you ever gotten mail that was misaddressed?
Yes.
Yes. Okay, it's not that hard.
So then what happened? Where is it showing us that happening with Scott Minnich's experiment? Like,
I don't see it. So that's a good question. Where is the promoter capture? See, I don't see something that looks like that in this diagram. And I don't know if he had it anywhere farther. So how about we
ask him that?
Yeah, you seem to be much more adept at keeping notes about and organize what we're going to ask. And so the first question is, can you help us understand this diagram? Are we understanding is that an MK or MA? And are we understanding? And then is that MK actually the promoter region? And then where is the promoter capture in the diagram? So that would be very helpful. But I think what's really important is, one, there are no new genes. There's absolutely no new genes.
Now, is it possible that the USPG is the promoter part that got, like, they did it two different ways, like the E. coli did it in two different ways, because that's what it seems to be showing here, that like it was two different pathways to get to that metabolizing citrate.
I think we're going to have to talk to Dr. Minnich.
Okay, we'll leave it at that.
We'll leave it. Okay. Well, this is the purpose of our discussion. We didn't want to. We wanted to have credible questions that will enlighten everyone. And I told you I was going to fumble through this.
Oh, this was amazing. Sal, you did an amazing job explaining these concepts. Thank you so much. I think this is a great show. I hope this will be helpful to people.
Well, see, this is where, people haven't seen both of us very often we're not on YouTube quite as much, because I'm now having conversations like this also privately with people. I'm learning so much. And there are things that you won't learn in the classroom because they have it structured. And then it's like, okay, I'm an engineer. It's kind of nice. I could talk to Dr. Minnich, who's a microbiologist. And like, I don't have to go through like 3 years of microbiology school to get some answers that could really be very helpful, and then vice versa. And by the way, this sort of highlights just how broad the field of biology is. Just when you think you know a lot, you may know a lot in one specialty, but then you know nothing in another specialty.
You
It's
know, well, yeah, I hope that people like who can just appreciate the complexity alone in one portion when you showed that Krebs cycle. I mean, we're talking about one system of many, many complex systems within every organism that exists. There's no such thing as a life form that is not incredibly complex. And so to me, this is clear, clear evidence that there is a mind, that there's an intelligence behind life. And so I hope people who have been in doubt about the creator would be able to see this and know that, yeah, there is someone who responsible for this.
so my purpose also in doing this stuff on Academic Channel, it's like, okay, this I think it's helpful for everyone to kind of just see how difficult it is to learn this stuff. And it's okay to fumble through this, make mistakes, and then formulate questions, and then talk to people who will actually be able to explain it to you. We're trying to show you the whole process. I do have some other comments. If you had anything you think I can help you with.
No, I think that's really good for right now. And I will do what Scott Minnich wanted us to do and study that Krebs cycle. So, yeah.
Well, Rebecca and I had some discussions like, well, how many of these shows are we going to do? I'm just like, well, I could envision 4. She's like, no, I don't think I want to go to 10. I mean, I'm just like, well, that is true. So I think we've come to, I mean, studying the Krebs cycle is going to be kind of funny because I did study it in biochemistry. I had to recapitulate it on a final exam. He was old school. He said, you're going to draw that and I'm going to see how many. I got it right, but it took me 11 hours of memorization to get all the names of all the parts. And I'm embarrassed to say, you asked a probing question, what does it do? And I'm just like, oh my goodness, I really don't know. I mean, what does it do for us? And I'm like, that's kind of bad. I was supposed to know this, and I don't have an answer. But it converts some. I think it converts some of the sugars or something to helps us make ATP or something, or pyruvate. And the biochemistry guys are going to just slaughter me because I didn't answer that correctly. My biochemistry teacher would be just kind of cringing, or several of them. But
Let's send it to Sygart and see what he says.
Yes. Oh my goodness. And he was a professor at my school. Now I'm really embarrassed. So see, this is what's also good when you start having these conversations, you have to be reacquainted. So now let me give you some of my closing thoughts.
Okay.
There is the first interpretation that bothered me in 2017, I'm like, this is very interesting that you could do this faster than Lenski. But my first reaction was, Dr. Minnich, I don't know that we've necessarily broken it, because you could show this could evolve so much faster. So you've actually helped the evolutionary case, because you've shown that it's really easy to evolve this. And therefore, we're just going to multiply Lenski's argument by that, many factors. You were able to do this 300 times faster than Lenski, or whatever factor. And now you've just kind of improved his argument, because you're showing E. coli can evolve. And I'm like, that does make me feel really, really uncomfortable. And I got the impression Dr. Dan was kind of citing, someone said in passing that Dr. Dan was really hammering Dr. Behe about Minnich's experiment, saying, look, this is way easier to evolve. And I'm like, okay, so I'd say Dr. Dan, I think has give credit where credit is due. That's kind of my sentiment too. It's like, well, how does this help the ID case? You've just shown that if Lenski were more competent or more lucky, he could have been evolving this way faster. But this is the way I look at it. I said there are definitely things, we're just like the cecal valve. They can evolve things new, but you can't extrapolate. If it's particularly easy to evolve, that doesn't mean you can evolve the hard things, because there are clearly things that don't evolve. There are clearly things that don't evolve, and the bonus is Lenski's experiment is losing genes, and I'm just like, people on our side of the argument that saw that when I gave them that title, they just shook their head and said, why do people believe this stuff? It's like, well, that's exactly what I was thinking. So my first thing to say is, well, it does speed it up, but is it relevant to the evolution of new genes? Because this is just, you're duplicating genes and you're rewiring the promoters, which is the switches on and off. That is trivial compared to being able to build a new gene family. And unless someone has actually worked with this a lot, it's hard to convey that to someone who hasn't. But I think Dr. Minnich's colleague could see this as a problem. I could tell you this is not. I can almost guarantee that Joe DeWeese, who's Mr. Topoisomerase in the ID community, will say gene duplication is not going to make a topoisomerase if it didn't pre-exist. You can't use a process of duplication to make a gene of that level of complexity. That would be just absurd, and I would have to agree. But it's like, unless you actually know the structure of real, novel proteins, to the outside world, this looks like, well, it's all the same thing. It's like, no, it's not. It's not. So that's the first thing I'd say. The second thing, there's something else I wanted to comment on. So I was concerned that it was adapting real fast. Oh, now I remember. I'm privy to something that I'm sure Dr. Minnich will share. He was saying these things won't survive in the wild. And I've been saying for a long time, Lenski's crowing about this new species he's created. If you throw it out in the wild, where it had to compete against bacteria, other bacteria, it's going to die. It's going to go extinct real fast. I'm like, I could
Why
see
would it go extinct if it was subject to other bacteria?
It's very fragile. Okay, well, not. It's lost a lot of ability. So, I think he said, okay, what Lenski evolved is like evolving poodles and chihuahuas, and if you throw it in an arena where there are coyotes and wolves and foxes, I mean, that's the analogy. So those are more capable of. So I would say it's the durability and ability to, real bacteria have a lot more in their arsenal to be able to cope with all these environments. This E. coli can only live in that environment. It cannot live in a real environment. It's so specialized. And obviously, if it's been losing genes, it's definitely specialized.
Well, to me, it's not really a valid criticism, because the whole point is to show the process of evolution and like how something can change within a selected environment. So what I'm trying to say is, that bacteria may not survive if we just throw it back out into another environment. But if it is making positive changes in the environment that it's in, then that's a valid, argument for the evolutionary side. Do you see what I'm saying?
I think you made a very good point there. So I that didn't occur to me. I do know that we got there's some chuckling when we talk about, like, okay, this is Chihuahua versus coyotes. I'm just like, well, that is kind of cute, but you have a point. So I think I don't know how to handle that, except that I mean, for me personally, okay, just at a personal level, like, to me, Lenski just created this artificial environment where you created a creature that it really isn't going to survive in a real environment. And we do want to talk about real environments. And I think we could actually say that in real environments, there are probably, we could imagine an E. coli evolving citrate utilization. We can, that's a good Scott Minnich question, because he did point out the bacteria strain that Lenski started with was already defective. It had a disrupted DCUS gene, and I don't know, he knows what the terms are for that. And it really is, like I said, that's the first big mistake. It's kind of embarrassing. He was crowing that this is, oh, now going back to Lenski's claim, this is all contingent. And I wish that word wasn't used because it's confusing.
Yeah.
And it's from Stephen Gould. But what it's saying is it depends on so many, there's only one road to Rome, so to speak. And it depended on all these sequence of events just happening just right. And it turns out there are many roads to Rome. Because what Scott Minnich evolved in those 47 trials, each of them is different, but it's getting the same outcome. This is kind of funny where you could have multiple recipes to end up with the same cake. In practice, that doesn't happen, but in the bacteria, it's like, well, you know, and what that tells you is this isn't that rare, and maybe it's not that complex, because there are many ways to make it. I can make a paperweight. I could take so many random objects in my house and make them a paperweight. It's not that difficult. And by way of analogy, there's so many ways to do this. It's like, well, did you really create something that was that spectacular if there's so many ways to do it? This is not much more than kind of trying to make a primitive hammer out of a variety of rocks. It's not that hard. And so this is a little bit, I think, in that regard, it does show how evolutionists will hype something that is really of no significance and pretend they've made some great discovery in vindicated evolutionary theory, and it's nothing of the sort.
And I think you said it, and I think Scott Minnich said it, that there's a limit to what can happen, right? Like there's a limit to the progress or change that an E. coli can have. And so, you know, they've been able to show this in repeated experiments, but, you know, where are the other capabilities that would need to evolve if we're going to go from prokaryote to eukaryote and to the diversity of life that we see? We need it to be able to break limits. We need it to be able to break through beyond this basic change. And that's what we don't see.
Well, you know, there's something I just observed in this. I mean, we can clear it with Dr. Minnich. All of this is just copying what already exists. Even that promoter capture is a copy.
Yeah.
And I think you had a good instinct. How can you really make something, you know, the kind of novelty you need is not copying what you already have. Maybe we could just, maybe that would be the bottom line. We'll say, Dr. Minnich, yeah, even I'm willing to grant this is a spectacular a spectacular adaptive capability. But this is done just by copying. You can't, one cannot extrapolate that you copied something and even copied it in quote unquote the wrong places. Therefore, that means you could build brand new genes with new capabilities of, like, say, collagen, helicase, polymerase, whatever. It's I think we could run that by him. I would like to just hear it from him if this is mostly just copying the same stuff. I mean, it is kind of interesting. You can have all these capabilities by copying a little bit more of this or that, but I don't think that can solve the origin of new genes.
Yeah. Now, although we've been kind of being critical of the Lenski experiment, I want to echo what Scott Minnich said in that talk, because he actually said this was a good experiment. Lenski's experiment was a good experiment. And we probably should have said this to the beginning of the show. If you want to show that evolution happens, this is the type of experiment that you should be doing. And so we commend, you know, Dr. Lenski for doing this experiment.
Yes.
And for, you know, showing these results, because, you know, this is the type of thing that needs to be done more. And, this is, yeah. So I don't want to hate too much on the Linsky
Yes,
experiment.
exactly.
Yeah.
Uh,
And Scott Minnich said the same thing. So,
One thing that was impressive, he said, okay, if you were to, like, say, try to analogize this or extrapolate it to humans, this would be like 1.3 million years of,
yeah.
and some people on our side of the argument said, well, look, they even in 1.3 million years, you haven't created a new gene equivalently, and 10 to the 11th power human beings.
Yeah.
This should be very concerning. um, and so we have these projects called orphan gene projects, where we could have thousands. I mean, if we evolve from a common ancestor, this is going to be very problematic. We actually don't know how many orphans of significance we have, but even a few would be very, very difficult to explain. So yes, this I do want to credit, and I thank you for reminding me. We should do more experiments like this. And if I may just do my Kent Hovind invitation here, it's like, oh, after 65,000 generations, it's still an E. coli. Maybe a question to ask is, Dr. Minnich, what architecturally, what defines an E. coli? Why is it not a Yersinia, the one that he specializes in? Or why isn't it,
Yeah. That's a
Salmonella?
great question.
How do we define it, architecturally? I mean, I can define a eukaryote versus a prokaryote in general terms. So
Right.
what defines an E. coli? I mean, why can I say after 65,000 generations, it's still an E. coli?
Yes.
So
That's a great question.
yeah.
Well, thanks so much,
Well,
Sal.
thank you.
This has been a great
Thank you,
talk.
Rebecca. I'll see you. I'm going to shut off the recording. I'll just
Sounds
meet with
good.
you backstage a little bit.
Yep.
Okay. So you're recording off?
Oh, yeah. I have to shut mine off too.